welcome to the 10th Neuro Talk Evening with Professor Christoph Herrmann from the University of Oldenburg, the Karolinska University of Oldenburg. Today's topic is modulation of brain activity and cognitive processes through transcranial alternating current stimulation to tACS. And yeah, just to give you a brief introduction, now in my slide switch, it just takes a little amount of time. Just to give you the slight introduction, as mentioned, he is professor at the University of Oldenburg, and he has a quite interdisciplinary background, considering his diploma in electrical engineering, his PhD in computer science, and then also still holding a habilitation in psychology. Previous to his position on Oldenburg, he has been at the Max Planck Institute for Neuropsychological Research, and he has also been the professor for biopsychology at the University of Magdeburg. His research focus currently is neural oscillations and cognition and perception using EEG. And notable work is modulation of brain activity with non-invasive methods, with tACS being one of them. In his current studies, he's focusing on enhancing audio perception and temporal resolution with the transcranial alternating current stimulation. And to slowly shift the topic to today's topic, ultimately, altering our brain's activity provides one of the most promising research areas, considering neuroscience as a research field and also considering it related to being related to health. But how exactly can we use this potential without severely damaging the brain or evoking effects that we rather wouldn't like to appear? And with this being said, I would like to end my part of the presentation and then hand over to you, Professor.
Yes, good evening. Let me just start my slides and hope that you can see them properly. Can you acknowledge that you see my slides? I see thumbs up. Very good. All right, so thank you for the introduction. I'm going to talk about the modulation of brain activity and in turn also cognitive processes through my favorite method, transcranial alternating current stimulation. And what I have in mind together with you today is, at first, I will give an introduction to the method, so about brain oscillations and why they're interesting for cognition. Then we can have a session of questions there. Then I will talk about how to modulate these EEG oscillations with tACS. I'll introduce to you what resonance and entrainment mean. I will talk about after effects of tACS in the EEG and the duration of those. And I will talk about the online effect of tACS. And then we can have another round of questions. And finally, I will introduce one experiment that we did, where we not only modulate brain oscillations, but also cognition, and that is mental rotation, as you'll see. Okay, so to begin with, why should you and am I interested in brain oscillations? Here is a very nice example. The participants have to sit in front of a screen and they watch a fixation cross in order to reduce the eye movements. And then for a very brief moment of time, 57 milliseconds, a letter appears on the screen, 1 out of 4 possible letters. And the subjects have to indicate by a button press which letter it is. And then there is a mask following that with all 4 letters. That makes it hard to perceive that. And some subjects perceive these letters in like 90% of the trials. We call them perceivers, and the others operate at chance level. We call them non-perceivers. And what I'm showing here are the EEG topographies before the onset of the letter for the 10 Hertz activity, the human EEG alpha activity. And you can see the perceivers have low amplitude alpha oscillations and the non-perceivers have high alpha amplitude. So that determines whether or not they can see these near threshold stimuli. This is between subjects, but that would also work within subjects. So that's one rationale why I think brain oscillations are interesting. And then we typically look at them with either EEG, so electroencephalography, or MEG, magnetoencephalography. And for those who are not familiar with the method, I wanted to give a brief introduction how a neuron, and I hope everybody knows what a neuron is, or neural activity leads to EEG activity. What you see on the slide is supposed to be a pyramidal cell P in the left of the picture. The surface of the cortex is in the top of the graph. And on the very top, you see how an EEG recording would look like. And then you see 4 intracranial recordings that look quite different or similar. So the input to the neuron that comes from, let's say, the thalamus, when it perceives a stimulus, are action potentials that they look quite different. And then if you look at the dendrites, where you have postsynaptic potentials, they actually look quite similar to the EEG. And then when you go further down in the recordings and look on the axon again, there you see action potentials that look different. So what the EEG of a region reflects is rather the input to the neurons there, rather than the output. And then just because I will also show some MEG data, what we see in the EEG are the extracellular currents that are shown in yellow in the right half of the blue diagram, while the current flow inside the neuron is the red arrow. And if you apply the right-hand rule, only works for the right hand, and point the thumb into the direction of the current, so down in this case, then you would see the magnetic field lines as they are indicated in the MEG picture. So the EEG field, the electric field and the magnetic field are rotated by 90 degrees. And therefore, if I have a dipole in the back of the brain, as shown in the lower left, then this would yield the topography in the middle in the EEG, with negativities in the back and positivities over the front, but a 90-degree rotated topography for the MEG. Red indicates where the field lines exit the brain, and blue is where they enter the brain again. Now, how does a brain oscillation come about if the neurons only fire action potentials leading to postsynaptic potentials? And we can look at that in simulations. The whole brain has a lot of inhibition in order not to get an epileptic seizure. Almost all pyramidal cells have inhibitory feedback, as shown here. So on the left, you see a pyramidal cell, and from the top, you see the input. And on the bottom, you would have firing output. And let's assume there is constant input coming from another neuron in case of input from the periphery or something. So as soon as the pyramidal cell starts to fire, it will excite the inhibitory cell, and the inhibitory cell will inhibit the input. Therefore, the pyramidal cell stops to fire, the inhibitory cell stops to inhibit, and the pyramidal cell starts firing again. And depending on the exact length of these axons and the time constants, you will get different patterns. But on the right, you see the firing of the pyramidal cell, where you always see 3 action potentials in a row, and then you see 3 inputs that do not reach the threshold because they are inhibited. And this is sort of the minimal neuronal oscillator that you can build. And there are even neurons that oscillate by themselves. But if you see that, you can imagine that depending on how neurons are interconnected, how long the axons are, whether the axons are myelinated, etc., the brain can generate oscillations of different frequencies, and the parameters of the neurons and the axonal connections determine the frequency of the oscillation. So this is a picture of a pyramidal cell and the inhibitory feedback. So these are very short and therefore fast connections in the range of micrometers to millimeters. So we have short time delays on the axons, and the whole loop, if you will, is rather quick. That leads to high frequencies such as gamma activity in the 30 to 90 hertz frequency range. However, if you have longer axonal connections, like going from the thalamus to the cortex, like from the lateral geniculate nucleus to V1, then you have connections on the order of centimeters, and they're myelinated. So There you have longer time delays on the axons that yields lower frequencies. And the model that I'm showing on the right that has even made it into Wikipedia by now is assumed to be the model of how the visual alpha activity is generated. And what I'm going to talk about a lot today are visual alpha oscillations in the EEG. And already Hans Berger in 1924 found out that they are important for or reflect the processing of the brain. And here's an example from visual processing. So over the course of time, the amplitude of alpha activity waxes and wanes, so goes up and down in amplitude. And when the amplitude is high, it is unlikely that one can perceive a near threshold stimulus. So let's say a brief stimulus like the P that we looked at. And if the alpha activity is low, then it's more likely that we perceive such near threshold stimuli. And just because I'm going to show that later on, this picture from Flavio Fröhlich also had a time frequency representation. You all know the time representation in the top graph. And in the bottom graph, you also have time as the horizontal axis, but then we have frequency of the vertical axis, and the color indicates whether a frequency is present in the data or not. So the three orange blobs represent these three bursts of alpha activity. And this all has led to a model of how alpha activity is important for visual perception. And this was shown here by Kyle Matthewson. But there are many nice papers that show that the alpha amplitude and the alpha phase play a role for visual perception. And in this review, he showed an example where he called it the power or the amplitude. I would say this is the phase here, but you see for those trials that were not detected, the amplitude was high at that moment. And for those trials that were detected, the amplitude was low. So it's a difference in phase basically. And they derived a model from that they called the pulsed inhibition model, where in A on the left side, you see the threshold, the dashed line, that the neurons that fire in response to a visual stimulus have to exceed in order to make visual cortex fire. And the white sinusoidal line is supposed to be the EEG or the LFP. And if the amplitude of the alpha oscillation is very high, like in part B, then you actually have regions of inhibition that are marked in red here, where even if a stimulus is presented, the postsynaptic potentials would not make the neuron fire. They don't exceed the threshold. Therefore, he says this is the cortical excitability along the y-axis. That is my introduction to the EEG. And I think Sebastian Wilhelm has planned that we have a first round of questions. Is that right?
Of course, if there are any questions by now. I mean, at least I had one, but I would let other people go first, just in case.
Don't be shy or don't bite.
But then maybe I just start, and then
Sure.
somebody feels encouraged. So by just screening the literature a little bit, I've seen a few papers claiming that the effects aren't that clear, and there's also around about 30% of non-responders, so people that don't get the effects. And I mean, by now we see a pretty clear relation, but is there anything known why they don't respond to the simulation of their brain activity while still having the normal behavioral effects with the alpha effects in the visual perception?
Are you asking about the effects of alpha on perception or the effect of tACS on alpha? That would be two different answers.
tACS on alpha.
tACS and alpha. Yes, we know a number of reasons why tACS cannot influence the EEG. The most prominent one is if the alpha activity is already very high, like when you have your eyes closed, that seems to represent a ceiling effect. The amplitude cannot go higher than that. We had that by accident in one of our experiments, and luckily continued experimenting and found out if we just let the people open their eyes, then we can still modulate the activity. But it also probably depends on proper planning of the experiment, on actually making sure with modeling, and I'll talk about that for a little bit, that enough current density reaches the target region, which is different for different subjects with different thicknesses of skulls and skin. But also we just published something that was not very conclusive, but already indicates that the tiredness of our subjects also plays a crucial role. And probably over the years, we'll find a bunch of further modulators.
Sorry.
Yes, there's a question in the chat, I guess.
Yeah.
And Alexia is asking, could you go a bit in depth on why a higher amplitude results in a lower excitability? What is the physiological reason behind it?
If you have a high EEG amplitude on the scalp, you also have high local field potentials inside the brain. And the membrane voltage is the difference measured from the inside with respect to the outside. And if the outside oscillates up and down, then of course also the membrane voltage oscillates up and down because its reference oscillates. And if the amplitude of the oscillation becomes bigger than a single postsynaptic potential, then during the negative half cycle, the action potentials, or sorry, the postsynaptic potentials can no longer exceed the threshold. But if you're in the positive half wave, they can exceed the threshold.
Thank you.
So there's one more question. Please, Alenka.
I hope you can hear me since I'm in the U-Bahn.
I can hear you.
Okay, awesome. So I think I missed it, or maybe not. Is there a specific place where it will stimulate in the brain?
I'll go into that now.
Okay, thank you.
Today I will only show visual experiments, and there we stimulate over the back of the brain, but I'll get to that.
Okay. Then I do think there are no more questions at the moment.
Okay.
Please go on, and we will have a second opportunity for questions just in a few minutes, 15 or so.
Right. All right. So now I want to talk about how to modulate brain oscillations. And in order to understand some of the things that I'm going to say, where I will use some physics talk, I want to update you to all be on the same page. And at first, let's look at a resonator. You probably know that I think every physical system can brought into oscillations. You know that from a wine glass. You either wet your finger and rub on the rim of the glass, like on the picture, and bring it to oscillate, and you can hear that, or you can also sing at the glass and even make it burst. So that is a resonator. And I don't call it an oscillator because it does not oscillate at rest. Nevertheless, it has a resonance frequency. So if you sing at the glass at different frequencies, you will see that the amplitude of the oscillation of the glass, is highest at its resonance frequency. That is also where it can break. But the resonator always resonates at the frequency at which you drive it. And therefore, this is not what we would call entrainment. And I'm going to argue that tACS entrains neurons. Now let's also look at an oscillator. And here we actually have two. So a very simple oscillator is a pendulum clock. And here are two pendulum clocks that actually entrain each other. That was demonstrated by Huygens, a Dutch researcher in, I forgot the year. And the big difference is the oscillator oscillates at rest, and therefore it has an eigenfrequency, even if you don't drive it from the outside. But it can either oscillate at its eigenfrequency if you leave it alone, or at the driving frequency. You can imagine that from a swing in the kindergarten, where you can just push the swing back and forth and enforce the frequency you wanted to show. So therefore here an entrainment is possible, and I will show you in a simulation why we assume that this is actually the mechanism when we look at the online effects of tACS. And in order to disentangle whether a system oscillates at its own frequency or is entrained to an exogenous rhythm, in physics there is a concept that is known as the Arnold tongue, and I'm going to go through that with you. So on the horizontal axis we have the frequency at which the system is driven from the outside, and on the vertical axis we have the intensity of the driving, so milliamps in case of tACS. And when you see the light gray regions, that's where the system shows its eigenfrequency and doesn't care about the driving. And where you see the dark gray color, that is the system entrains to the external force. On the right hand, you can see what that means for the tACS wave and the intracranial firing. But the interesting thing is that in this case, the eigenfrequency is at 10 Hertz, and you can see that if we also stimulate the system exactly at its eigenfrequency at 10 Hertz, then we need very, very low intensity to actually entrain the system. And if we increase the intensity, then we can even entrain over a wider range of frequencies. So where I have my arrow right now, that would mean that, let me go maybe here, I can entrain the system all the way from 9 Hertz to 11 Hertz. So the ongoing frequency, the eigenfrequency that wants to oscillate at 10 Hertz can be shifted down towards 9 Hertz or up towards 11 Hertz. After that, if I go to 12 Hertz or something, then it jumps back to its eigenfrequency. This concept is called the Arnold tongue, and it's nicely described in the book by Pikowski and co-authors. And what we did, try to look at a artificial neural network, a network of spiking neurons, where we connected 800 excitatory neurons with 200 inhibitory neurons in a fashion that is shown in B, where we have many short axons with fast delays or short propagation delays, and fewer long axons with long propagation delays. And if you look at C, and if you look at the activity of the system, you can see that it shows a oscillation even when it is not driven. And in D, you just see the single spikes as dots of all the different neurons. Blue are the inhibitory ones and black are the excitatory ones. And if you average that, you see this black pattern at the bottom. And now the nice thing is with this neural network, I can do something that I won't dare to do to my human participants, because now I can try many different simulation frequencies, in this case 1 through 100 Hertz, and also many different intensities. And then in graph A, we plotted the power that we observed for these network oscillations, and in C, we plotted the frequencies. And at first in A, we can see, we do see these Arnold tongues, one at the eigenfrequency that we set with the connections, but then also a 2-to-1 Arnold tongue at 20 Hertz, a 3-to-1 Arnold tongue at 30 Hertz, and a little bit weaker Arnold tongue at 40 Hertz, and maybe even at 60 Hertz. And interestingly, in graph C, the black lines don't belong to the data. That's just to indicate roughly where such an Arnold tongue can be observed in A. And here we show the frequency, and you can see for high intensities, I go there with the mouse again, you can see that the frequency increases all the way from roughly 5 Hertz to about 15 Hertz. And then before it goes into the next Arnold tongue, it jumps back to a lower frequency. So these are exactly the phenomena that are described in physics if a system undergoes entrainment. And therefore, and due to some animal studies by other people, we think that is what is happening during stimulation. And I like to say, always simulate before you stimulate, because we need to know where in the brain we stimulate. And we like to do that with a so-called finite element model derived from MRI. So we automatically, what do you call that? We do that in FreeSurfer. You classify all voxels as either being skin or skull or liquor or gray matter or white matter, and they all get different conductivities. And then we also stick electrodes on the head, shown here in black, and then we apply a current to flow through the skull and the brain. And the colors in the brain indicate the current density in these regions. And then you can do two things. You can look at the pattern and make sure you're stimulating the visual cortex or auditory cortex or what have you. And also you can go there and look at the voltage gradient, because it's known from animal studies that you need at least 0.2, better 0.3 volts per meter to actually modulate the firing of the neurons. And then you only modulate the time of the firing, but not the absolute firing rate. But if you do that, you can learn a lot of things, and I'm trying to show that here. So in this study, we recorded the MEG shown in the top right. We looked at the MR images of all subjects. We derived these head models. And now on the bottom, you see two nice things. We localized the generator of the alpha activity of the individual alpha frequency for every subject. And here it's shown for one subject. And you see, yes, it's over the occipital cortex. So that's, let me go there with my arrow, this image. But now from our finite element simulations, in the same space of the brain, we know where in this very subject we will actually have current flow. And the intensity of that and the pattern of that will vary from subject to subject. And with our linear regression, we were able to demonstrate that a good correlation between these two measures actually gives us a higher effect. If for some reason the pattern is too weak because the skull is too thick, or it's in a different region for whatever reason, then this would have not been significant. And that was tested separately for the two conditions. Here you see the correlation for the stimulation condition and for the sham condition. And you can see for the simulation, the dots for power observed and power predicted are nicely along a line. And for sham, it's a region around 0. So we were very happy about that. And now we did one more simulation for the after effect. That was a question that came out, I don't even remember, I think it was from the reviewers in our first paper in 2010. They said, well, the effect that you observe, I'm going to show that later, is after the end of the stimulation. So how does that come about? And the best thing we could think about is that it is due to synaptic plasticity that depends on the frequency of the oscillation. And let me go through this with you slowly, because maybe it's complicated. Regarding the synapse between the presynaptic and the postsynaptic potential, there can be different cases whenever the postsynaptic neuron fires. You can look at two conditions. Let's start with the right one. Here, the presynaptic neuron fired first, and then the postsynaptic neuron fired. So this could have been causal. And in that case, the synaptic strength is increased. That has been observed in animal recordings. And the closer the timing, the stronger the change of the synapse. If, however, what's shown on the left, at first the postsynaptic neuron fires due to activity from some other neurons, and then this very presynaptic neuron fires, that is not causal, and then the synaptic strength is weakened, which is referred to as long-term depression as compared to long-term potentiation. And we made a very simple neural network simulation where we had one neuron shown in gray that is stimulated with a 10 hertz spike train. And then we had multiple different neurons that were connected by axons of different lengths, and therefore they had different turnaround times, shown here for 80 milliseconds corresponding to 12 hertz, 100 milliseconds corresponding to 10 hertz, and 120 milliseconds corresponding to 8 hertz. Now, all of the synaptic strengths were randomly assigned a value, which are shown as gray dots here. And then we started to apply 10 hertz simulation and train the weights according to this spike timing dependent plasticity rule. And guess what happened? You, of course, see the pattern of the thing that you have over here, but you don't see the blue part because it doesn't go below 0. But you can see that the black dots represent the synaptic strengths after the end of this training. And then we said, okay, on average, the synaptic weight was 4 before stimulation. And wherever the synaptic strength was stronger than 4, we refer to that as LTP. And wherever it was below that, shown here in blue in the diagram, would be LTD. Now note that we stimulated at 10 hertz, and the region here is from 80 to 100 milliseconds. So that is the region from 10 to 12 hertz. So the simulation, if the stimulation frequency is a bit faster than the turnaround of the neuronal loops, that is ideal for the after effect. So that brings us to an intermediate summary. In order to achieve an online effect, which is a bit harder to observe, but I'll get to that, it's most effective if the driving frequency is exactly at the eigenfrequency of the system, so the individual alpha frequency, for example, or if the driving frequency is any n to m ratio where n and m are integers multiplied with the eigenfrequency, so 20, 30, or other weirder numbers. However, for the offline effect, it's most effective if the driving frequency is a little lower than the eigenfrequency. Lower? Did I say that wrong just now? A little faster. The sign is wrong. Sorry for that. And with that, I'm going to go to some physiological experiments where we actually stimulated the people over the back of the head and at the same time looked at the EEG. But in this case, we were not able to look at the EEG during stimulation because we are stimulating on the scalp with voltages in the range of volts. And we are recording EEG very close by, where we record voltages on the order of microvolts, so a million times smaller. And even though we know exactly what stimulation sign we used, it's very hard for software to subtract that, and it's tricky, but we found a way to get rid of it. So the experiment went as follows. At first, we detected the individual alpha frequency, which varies for every subject, of course. And then we detected 2 thresholds, the sensation threshold for the skin, where you feel a tickling sensation, a bit like touching the poles of a battery with your tongue. We did that as kids in order to find out why the toys won't work anymore. And the other is the threshold for perceiving a flickering sensation in the periphery of your visual field. Both of them would give away to the participants whether they are in the stimulation block or in the sham block. Then we record the EEG before stimulation. Then we stimulate for 10 seconds, either with tACS or with a make-believe sham condition. And then we record another 3 minutes of EEG. And luckily, in the group that got stimulated, we see an elevation of the spectrum from before to after simulation. And this is averaged across the subjects and washes out a little bit. For the stats, we took the values from the individual alpha frequency. And for the sham group, you don't see a significant difference here, even though descriptively it goes in the same direction. But you can see in the stats that was clearly significant, and we were very happy to convince our reviewers that there is a plausible cause that can be assumed for this after effect. And then the paper got published. We only looked during that experiment at the first 3 minutes after stimulation. And of course, that was something that was interesting to us and will be interesting in the future when we and others try to apply that maybe as a therapy in diseases where oscillations are malfunctioning and need to be restored. What is the duration of the after effect? So we did a similar experiment and asked our subjects to stay there for 90 minutes after the end of 20 minutes of stimulation. And they again had a very boring task. We call that a vigilance task, where the fixation cross changes orientation every now and then, and that has to be detected by a button press, because if you sit in a dimly lit chamber and don't do anything for 90 minutes or a whole hour, if you take the time together, then some people fall asleep and don't react, and the EEG completely changes. And then in this graph, we took the amplitude of the spectrum of the individual alpha peak and started plotting that after the end of the experiment. And what we expected was that it would be elevated in the beginning and that it would decay over time until it's no longer different from the sham group. But that's not what we found. Instead, we found that it stays elevated and it even ramps up further, but so does the alpha activity in the sham group. And this is something that has been described. If you just sit in the lab for a long amount of time, the alpha activity ramps up, and so does the error rate and the reaction time. And after 70 minutes or so, the difference was no longer significant because the sham group also reached high levels of alpha activity. But if you look at the two spectra or two groups of spectra in the lower right, you can see that for stimulation, the increase of alpha was much higher and actually follows the time. So it really goes up over time. And the reason why we need a sham group always is that also in the group that does not receive stimulation, the alpha activity will be bigger after stimulation as compared to before the stimulation. But the difference between these two groups has to be significant. And then we did succeed in looking at the online effect also in EEG, but others tried to replicate that and were not able to replicate that and said they don't believe it. And after some time, we actually got our own MEG, and that is advantageous for looking at the effects of tACS, because here the sensors do not touch the skin. We have volts of current, volts of voltage in the scalp, but not in the MEG sensors, and therefore the size of the artifact is already considerably slower. But still, if you just do something that we did here, we presented visual and auditory stimuli and looked at the P1M and N1M and the topography, and you don't see an auditory and a visual topography, but you see the two wires of tACS leading to the OZ stimulation electrode and CZ stimulation electrode. But a beamformer actually extracts all the activity that comes from outside the head or separates it from the activity that comes from inside the head. And therefore, with just applying the beamformer once, you completely get rid of the artifact in a very nice way. So let me talk you through this slide. In the top left, you see the sham condition. And the two spectra are for the eyes closed with higher alpha and eyes open with lower alpha. You see the topography of that. And after the beamforming, this is being transformed into the brain. And you can see by the blue activity over here that this is nicely located to the occipital lobe. And in the tACS condition, you can see that there is no difference between eyes open and eyes closed because you only see the artifact. This is much stronger than over here. But after the beamforming, and you also see in the topography that the topography has nothing to do with brain activity. If something comes from outside the brain and goes through the brain, then it has a flat topography. But if it comes from inside and the magnetic field lines leave the head, then you would see positive and negative patterns. You don't see that here. But after beamforming, you see the same source localization as during sham when there was no stimulation. And also you see the spectra differentiate between eyes open and eyes closed. And you can see that both peaks are elevated as compared to the sham condition. So that was very nice and made us hope that we can also do experiments where we have some more interesting task than our vigilance task. But again, I'm giving you the chance to ask some questions in between.
So, thank you again. And as Professor Herrmann mentioned, please feel free to ask anything you would like or you're interested in. I mean, right after we started with the second part, a question came up in my mind. So while the others might consider how they formulate their questions, could you model the reactivity with single compartment models?
I would love to have a more fine-grained model of the brain, and especially I would like to model also the synaptic connections in order to see in a model we can just switch them on and off and change the timing and see how that affects the after effects and the online effect. But I work in a psychology lab, and most of my employees are psychologists or neuroscientists and not so skilled in doing these complicated network models. So the network papers that we did were always done with people from outside the lab. And many modelers seem to have a fable, or they favor single formula simulations of a network instead of having every single neuron modeled separately. About single compartment, yeah, of course, you couldn't go to any level of detail. There is a paper by Flavio Fröhlich where I think he used Hodgkin-Huxley neurons and modeled all the details of them.
So, thank you.
If somebody wants to do that, contact me and maybe you can do your master's degree or your PhD on that topic.
So there are two more questions. I will start by reading out the one in the chat, and then Mr. Wang can ask his question.
Okay.
So, they've both been written in the chat.
Yes, I just can I ask the question now?
Give me a second.
Okay.
There's one question from Alexia. She asked, what is beamforming? I guess you mentioned it, but could you please repeat it?
I only mentioned roughly how it works. I'm not a mathematician. I think it uses a Taylor-Reihe. What is that in English?
Taylor series.
A Taylor series in order to separate the activity that comes from inside the head from that comes from outside the head. But I cannot tell you how the math works behind that.
I hope that answers the question sufficiently. I don't think we can go
Yeah.
any deeper. Okay. Or would you like to do a follow-up?
you.
Okay.
No,
Mr.
no,
Wang?
I'm good.
Thank you very much. Hello, Christoph. So my question is actually is based on this alpha, so the effect on alpha. So maybe I just missed some details. So my actually really interesting is actually we if we look at the alpha power, right? It's especially so the eye open and eye close. So I see there's significant increase the difference between the eye open and eye close, right? And then the effect is actually overall is like it's only effect until the dynamics change. So like doing task or not doing tasks, I just reference is to eye open or close, right? So we can also thinking is like in the visual discrimination tasks is like okay, I'm finding the overlapped masked things, or I cannot find it, is something like, okay, this alpha is actually reference to the dynamic change to the high and low, or this is actually overall. So in the baseline, the alpha is increased, and in task, the alpha is also changed, but the dynamic is also changed. So actually, in what case is it?
In the experiment that I'm going to show, we're actually, yeah, that touches on that, but it doesn't answer it. Can we answer that with a nice experiment? We know from experiments that I didn't show that if the alpha activity is very high to begin with, then we cannot elevate it further. So if you have your eyes closed, the alpha seems to go into its maximum and does not respond to further tACS stimulation. Also, if we have subjects that for some reason, even though they have their eyes open, just have a very high alpha activity, we have to exclude them from our analysis. So we just take them as outliers if they have more than, I don't know, 2 standard deviations of the alpha of all the subjects of this experiment, and we exclude them.
It's actually very interesting. It means that there's some like a upper boundary for the effect, right? If the alpha is already very high, even I bring the tACS, I cannot elevate anymore.
Well, we're not sure whether it's a ceiling effect. There is another potential explanation. Of course, we stimulate with a maximum intensity, because if we increase the intensity further than, say, 2 milliamps peak to peak, then you really feel the stimulation, and that would give away whether you're in the sham condition or not. But also, if we go up to 4 milliamps, that's where the subjects say, stop, we don't go any further. And some labs actually apply, what's the name of the cream, ESLA cream or something like that, to the skin beforehand, but that has to be done by medical personnel. We're not allowed to do that in a psychology lab without having a doctor there. And then you have to put the cream on, let it sit there for half an hour, cover the skin with foil so the cream doesn't, I don't know, evaporate. That's quite complicated, and we haven't done it so far. Then you can go even to higher intensities, and maybe then you could increase also the closed eye alpha.
Thank you very much.
You're welcome.
So, also thank you from my side. By now, I don't see any more questions. I had one more, but would you like to go on now and ask me later, or me asking you later, or may I just ask?
Well, if it's about what I said so far, then you might as well ask right now. If it's very general, you can wait till later. I don't care.
It's referring to a question that has been submitted before. You mentioned...
I'm going to address those anyway.
Oh, okay, then perfect. Then
at the very end. Okay, then I continue. So now we want to do an experiment with a slightly more interesting task than our vigilance task, and that is ventral rotation. Many of you will probably know this task because it's very well known in psychology. Participants are shown 2 objects, and they're supposed to say whether they are same or different, but they can be different, they can be the same and rotated. So if the rotation angle is very small, then it's very easy to do the task, and the reaction time is slow and the error rate is slow. But here you see an object in the top graph where the object is rotated 150 degrees, and it's the same object. So here you would make slightly more errors, and it would take you longer to do that. On the bottom, you see 2 different geometrical objects where you have to say no. So at large rotation angles, you get higher error rate and longer reaction times. The reason why this is interesting to apply tACS is that in 1994, Christoph Michel from Lausanne demonstrated the EEG and MEG responses to that, and especially the event-related desynchronization. So the alpha activity is highest when you have your eyes closed. It is lower when you open your eyes. And even during eyes open, it is further decreased in amplitude when you present a visual stimulus. And that is shown here in the graph and is called event-related desynchronization. And you see that for every visual stimulus that you present that I've seen so far. And here you can see that for these two different conditions, easy and hard, so big and small rotation angle, the alpha desynchronization remains present for longer in the hard condition. So we thought, well, maybe that is something where the alpha activity can be modulated by us and give us performance differences, because this is a purely correlational studies. They saw that the duration of ERD correlates with the performance. And before I come to how we generated our hypothesis, I have to go back to an old study of Byung-Yong Min in my lab, where he looked at the alpha activity of subjects, and it had a completely different intention. But we separated the participants depending on the amplitude of the alpha activity before a visual stimulus comes on. So the red line represents high alpha group, the blue line represents the medium alpha group, and the green is the low alpha group. And you can see that for the high alpha, there is also a strong ERD that is also long lasting as compared to the medium and the low. The low, there is hardly any desynchronization. There's only a little bit of synchronization in the beginning. So what we thought is, if we stimulate alpha all the time, irrespective of the stimulation, then probably the alpha activity is amplified in the baseline. So we move the people to this red condition, and that allows for a stronger ERD as compared to if they have low alpha in the baseline. And if that would actually work, then this could result in enhanced performance. And the neat thing would be that this is a causal study. We modulate something as the experimenter, and we see whether this has an effect on the performance of the subjects. And this is what Florian Kasten did in a rather lengthy experiment. So we recorded the brain activity again and determined the individual alpha frequency, IAF, measured our baselines, and then we had a baseline block where the subjects had their brain activity recorded, and they already did the first block of mental rotation to also have a baseline performance. Then during blocks 2 and 3, they were stimulated for 20 minutes or were in the sham condition. And then they had another 5 blocks after the end of stimulation, where we were able to look at both the behavior and the brain activity because it's not distorted by the artifact. The stimulation electrodes were OZ and CZ, just like the previous examples for which we also showed you the finite element model. And this is the time course of the mental rotation task. And this is the time frequency representation of the EEG in response to the visual stimulus, at first in the baseline. So let me go through this again. On the horizontal axis, we have the time in seconds. At 0, the stimulus comes on. And you can see that at 10 Hertz, you get this blue band here. That is the alpha desynchronization. So the 2 graphs that I showed on the previous 2 slides are basically a line taken out of this diagram at 10 Hertz, or at the individual alpha frequency. And here the line is just shown color-coded. So it's high before visual stimulation, and it goes down for visual stimulation. And now I chose blocks 4 and 5, that's after the stimulation. And I compared now the stimulation to the sham group, and you can already see it with the bare eye that the blue is stronger in the stimulation blocks than in the sham blocks. And statistically, we plotted here on the right how big the ERD was. So during the stimulation, it went to like 200% reduction. 200% can't be. I forgot what the axis is. And in the sham group, it remained 100%. And we didn't test that, but if you just look at it descriptively, it also looks like the blue region is shorter in time. So only like 2 seconds in the sham group and something like 3 seconds in the stimulation group. So we achieved what we wanted physiologically, and now let's see how that influences the performance in the task. So here we look at the change of the performance. So that is not the reaction time, but the accuracy. And you can clearly see that it went up significantly in stimulation. And if we look at the time course, so we said during the baseline, we consider that 0. And then during the stimulation, what's shown here in gray for blocks 2 and 3, you can see that immediately there is this increase in performance for the simulation as compared to the sham group. And then even after you switch off the stimulation, you still see this for the rest until block number 8. And that was something like 90 minutes or so. So for quite some time after the end of the stimulation. So that was very nice. We were able to modulate the alpha activity and its desynchronization, and that in turn led to a change in behavior. And with that, I'm at the end of what I wanted to say, and I thank you for your attention and my group dressed here in their Christmas clothing for the nice work that they did. And again, I'm happy to take questions, or shall I answer the questions that you sent me via email first? You decide the sequence.
I would say we can just start by answering the questions that have been submitted, and maybe the people just need a little time to process the last input, and then if a question comes up, please feel free to either by the signs you can use on Zoom, raise your hand or just write your question in the chat, and then we will intervene and tell you if there's any question.
And maybe if I misunderstood the question, the asker is here and can correct me. So the first question that I got via email was, how can tACS distort or enhance perception? And I tried to cover that in the beginning by introducing the pulsed inhibition model. And the next question was, could tACS be used to make learning phases more efficient? And yes, that can be done. We had one study where we looked at learning by Johannes Voskuhl, where we tried to make use of the notion that the number of gamma cycles that fit into one theta cycle determines the number of objects that you can keep in short-term memory. So we modulated the frequency of the theta activity down so more alpha cycles would fit in, and that actually did increase the memory in that experiment. But there are many other experiments where also the multiple cortices were stimulated at the same time with gamma here and theta there. And one group does a lot of experiments during sleep where they try to play the EEG that would be expected in certain sleep stages that are known to be relevant for memory, etc., etc. It's nothing that we are experts on, but other labs do that. Then from this, I can see that your group of listeners is already educated on what I was going to say and that they know the literature. So this person is asking, recent primate data suggests that if an endogenous oscillation is too strong, tACS may actually compete with it rather than entrain it. Does this suggest that we should avoid stimulating at the exact peak individual alpha frequency in healthy subjects and instead target slightly weaker oscillatory states to get better entrainment? And I have to move this away here again. How do I do that? Like that. So we never compared that directly because in one experiment we stimulated with 10 hertz and in others with individual alpha frequency. But at a descriptive level, at least, it looks like the individual alpha stimulation gives you bigger after effects than the 10 hertz. but there is also a ceiling, as I said before. And then we did one experiment where we tried to have two conditions. One is fixed 10 Hertz, tACS, and the other is an interleaved closed loop approach where we recorded EEG for some time to determine the individual alpha frequency at this moment, and then stimulated for some time again and did this on and on. And we're expecting that if we always stimulate at the individual alpha frequency, we would get the strongest after effect, but the opposite was the case. So if we stimulate with 10 Hertz all the time, that seems to be better for synaptic plasticity because you always stimulate the same loop as compared to jumping between loops all the time and stimulating every loop only for a fraction of the time. But probably if we would look at the alpha activity during the stimulation, then yes, stimulating at the individual alpha frequency would be probably best. This was shown by Heiko Stecher in 21.
I'm sorry, Professor Herrmann, before you go on, there are two questions in the chat. I just will start by reading out. So Mr. Wong, Dr. Wong, is detecting individual alpha very critical for such cognition modulation experiment? How sensitive will it be?
Actually, we had a study that never happened where somebody asked me, since I have never directly compared that, he would suggest that he does that and directly compare stimulating people simply at 10 Hertz with compared to individual alpha frequency. So I think it's an interesting question that still needs to be answered. And I expect that there will be a difference, but I don't know how big it will be. So how significant or important that is, I don't know.
Thank you. And then there is a second question from Alexia. Do the enhancing effects of tACS extend to all the modalities?
And with modality, you mean like auditory, visual, somatosensory, motor, etc.?
Yeah.
Yeah, okay. We don't know that because we have only done auditory and visual so far. And it is assumed that the other cortices, like motor and somatosensory cortex, favor different frequencies. And probably there you have to apply different frequencies. But I would say that apart from a different frequency, everything else should work similarly. And there are other groups that also do tACS in the somatosensory cortex and apply to the motor cortex, where I think they use beta and gamma more than alpha. Of course, there is also this arcade-like motor mu rhythm with this, which is roughly in the 10 Hertz range, but it's slightly different. It's not sinusoidal.
Makes sense. Thank you.
You're welcome. All right, then I'll continue on the next slide. So, yeah.
Sorry, just at that, there was a hand up for a second, but it was down again. Would you like to ask your question, or shall we just go on? So maybe the person can write in the chat and just. Oh yeah. In a few seconds, he will ask again. So we can go on.
So I'll go on with the email questions. Based on the literature, is it possible that tACS effects are cumulative and require long observation windows, minutes rather than seconds, to become statistically visible? Yes, absolutely. You can stimulate only for 1 or a few seconds and record the action potentials in animals, looking at the online effect. And then immediately when you stimulate, you see the effect, how the action potentials align to the sine wave, where you see them in the peaks, but not in the troughs. But I have already asked the animal experimenters in reviews that they please also try to stimulate for longer and to look at after effects, because at least for us, when we try to stimulate only for a very brief amount of time, like I think it was a second, we saw absolutely no after effects. And there is a paper by Andrea Anta where she did that for tACS and demonstrated nicely that I think she had to stimulate for at least 5 minutes to get an after effect at all. And she stimulated to up to, I don't know, maybe 20 minutes and was able to show that the after effect of tDCS on TMS-evoked motor potentials is longer the longer you stimulate. So if you look at the after effect, then yes, more than 10 minutes probably. And the next question is, most of your work focuses on the cortex, visual auditory. However, new data show motor learning effects from cerebellar tACS at theta frequencies. Should we begin to view the tACS as a tool for modulating, he says cerebrocortical loops, but maybe just non-cortical, non-purely cortical loops? And yes, I do think so. And we also have started to target different brain regions also in diseases. So the basal ganglia in Parkinson's patients and the anterior cingulate cortex in stroke. No, there were also Parkinson's patients. But there we use temporal interference stimulation where you have two pairs of electrodes that induce two patterns of current flow, and where they overlap, you get an amplitude modulation at your desired frequency, and everywhere else you just have a very high frequency that hopefully does not do anything to the neurons. So the problem of tACS is you can stimulate also deep regions in the brain, but of course you at the same time stimulate everything that's more superficial even more strongly. So it's not ideally suited to stimulate something in the depths of the brain. But with TIS, that is feasible. Questions, or shall I go to, I think, the last page?
By now, there is nobody raising their hands, so I think we can go on.
Then we come to the last but one question. In closed loop tACS, we try to stimulate in phase with the brain. But given the hardware and physiological conduction delays, are we ever truly stimulating in real time, or are we constantly chasing a past brain state? Are there any computational solutions by now, e.g. predictive coding, that provide a promising solution? That is, again, a very good question. And I have a postdoc from electronical engineering who is trying to build a closed loop system that is not that interleaved thing that I mentioned before, and we have not achieved it yet. But the speed is actually not the biggest problem. If you have a decently fast hardware and you are a good coder and can really do real-time coding, then you can do all the computations in a few milliseconds. So that's not the big delay. But you really have to make sure that you don't read in the whole data every time for 1 second that you want to analyze, but you only kick out one data sample from your ring buffer and enter a new one, and you just move pointers in the ring buffer, so everything is really fast. That is not the problem, but the problem that we have, and therefore he is trying new phase prediction algorithms, is that the phase prediction that we make for the near future quickly runs out of validity because the frequency varies. As somebody else already said, the alpha frequency, of course, always changes in frequency. And when you estimated the frequency, the phase in 100 milliseconds, that is, I think, the most important method that we have to solve. And then I'll come to the last question. Since the brain's alpha frequency is not static but drifts, e.g. to fatigue, don't fixed frequency tACS eventually becomes out of sync after 10 minutes? Shouldn't future protocols be adaptive by changing frequency in real time? And yes, again, I completely agree. For the online effect, that is probably better. But as we showed in our interleaved closed loop thing, for the after effect, it's better if you just stimulate with a fixed frequency all the time. And that is the end of the email questions.
Thank you very much. And there's the question where I saw the hand raised before. Please, Patrick.
I wanted to ask if it could be possible to just inhibit the alpha frequency of one hemisphere, or like are there some technical stuff that makes it possible? I don't know.
I like the idea, but I think what you're suggesting is that in one hemisphere we record the EEG, and in the other hemisphere we stimulate the brain with the hope that the artifact only affects the hemisphere where we stimulate. Is that the rationale of your question or proposal?
No, I just wanted to know if we could just like change the alpha frequency in one hemisphere more strongly and see like hemisphere differences with like, for example, if you would present something only on the visual field of the
Okay, yes, that's also a nice idea. Yes, that should work. So we always use electrodes CZ and OZ along the midline, but you could move the electrodes towards one of the two hemispheres and use something like, now I'm in the right hemisphere, so that would be O2 and C4. Then the pattern of current flow should be predominantly in the right hemisphere and should not affect the left hemisphere as strongly. And yes, then you could do visual experiments that stimulate the hemispheres differently visually. Nice idea.
And there is one more question. Please, Dr. Wang.
Chris, I think this question is actually a follow-up questions about this online real online in-phase stimulation, right? Based on the Pascal phase, the theory is actually we have the alpha oscillation or whatever, this low frequency, definitely some kind of low frequency. So we have this phase. The indicate is like, okay, I'm excitable or I'm inhibitory, right? So I'm in the excitable phase or I'm in the inhibitory phase. And now I'm not sure. So maybe I just missing the details about the tACS simulation. So actually, do we actually looking at the different phase? It's actually, you'll see we can have a lot of 10 hertz, right? We have a lot of 10 hertz of stimulation. And then we will have a different phase difference between my brain alpha 10 hertz and the stimulation 10 hertz, right? Have you ever looking at this phase where exactly which phase will give the stimulation?
We have not. We were always assuming. I mean, of course, if we stimulate from OZ to CZ, we have different phases, 180 degrees out of phase stimulation. And at least for physical systems, the phase follows the external driver. And we always assumed that that is the case, so that the phase of the alpha oscillation underneath the stimulation electrode would be roughly in phase with the phase of the stimulation sine wave. But we never looked at that. But it's something that you are completely right. There may well be a small phase shift, and that would be interesting to look at.
This could be, it's like if we have this external driver, and then we force this internal phase just change its phase, so to pass to your external, or just maybe we have different degree of the stimulation. So it's there some will work and some will not, right? So if you have from 1 to 108 degree, they will have something will definitely hit exactly the phase.
I think in the physical theory of entrainment, what follows the driver first is the phase. And it's harder to change the amplitude and the frequency. And since we think we're able to change the frequency, I didn't show that today, and the amplitude, I showed some evidence for that, we always assumed that the phase follows our simulation, but we never looked.
Thank you.
You're welcome. And they're very good questions, I must say. I'm positively surprised.
Thank you very much. I don't think there's one more question from Alexia.
Not really a question, but I just want to say to Professor Christoph, I really enjoyed your talk, and we are actually having a course on NIBS, and I'm actually reading your papers, and I made the connection earlier that I'm actually seeing a person that I'm reading the papers of, and I was really impressed. So thank you very much for the talk, and thank you for the organizers as well.
Yes, thanks for the praise, and thank you again. You're a very well-informed audience.
Thank you very much. And then there is one more question. Please, Leon.
Yeah, hi. I hope you can hear me.
Yes.
To give a counterexample for somebody who's not informed, that would be me. I'm more from the area of clinical psychology. And even though I do like neuroscience and I'm more involved in biological psychology, you mentioned that you are looking into certain diseases which could be treated. You mentioned Parkinson's as an obvious choice. Are there any further developments you are working on right now or in the literature, where I have to admit I didn't look into before the lecture today? But anything where you could just send me on my way to maybe look further into?
Yes, we just had a review on that, in which diseases in neurology and psychiatry, tACS has already been applied. And right now I don't recall the first author's name, but if you just search for my name and tACS and disease, it will pop up, and there we have a whole list. So it's a number of diseases already. So basically, whenever the disease is characterized by a change of a brain oscillation, then it's probably worthwhile trying. However, I have to say, it's easier to increase the amplitude of an oscillation with tACS, because in order to decrease it, you either have to work via the antagonism of two frequencies, or you have to stimulate with 180 degrees phase delay, which we are not able to do right now.
Okay, thank you. And maybe one more question is regarding to, I'm more inclined with the concept of RTMS, the magnetic modulation, just because I couldn't really decipher the differences. Well, obviously it's magnetic versus electron based, but from a psychiatric point of view, would that account for the exact system you just mentioned, that TMS is rather for network changes, and your system is regarded to more specific, let's say, procedures?
Another good question, and there are many differences between TMS and tACS that could be mentioned. So what's advantageous about TMS is it is a super threshold method. You can directly make silent neurons fire. You can easily demonstrate that. What we also do in our lab courses, you stimulate the motor cortex and you make muscles of the hand twitch. And that is, of course, much better because you can see effects directly, where the tACS is rather modulatory. So there has to be a brain oscillation in the first place, and then you can modulate its frequency and amplitude and probably also phase. So it's much weaker. But the advantage of tACS is you can tune it to a frequency, and then you only stimulate exactly that frequency. With TMLs, you have a brief impulse that goes through all frequencies mathematically. Of course, you can have a repetition rate that is 100 milliseconds, and that corresponds to 10 hertz. But then if you look at that in a time frequency diagram, you have the 10 hertz repetition as a horizontal line, but every 100 milliseconds, you have a vertical line through all frequencies. So you're never completely sure whether the effects that you see are because of the 10 hertz repetition rate or because every single impulse stimulates all possible frequencies. And another advantage of TMS is the disturbance of the EEG is shorter, and there are a lot of algorithms that get rid of them so that only a few milliseconds have to be thrown away. And after a brief amount of time, you can look at the EEG again. That's an advantage again of TMS.
Okay, thank you very much.
You're welcome.
Okay. And so now I came up with the question, way to answer the question about diseases. So Julian Keil, you may know him, you may not, is working with neuromodulation, so steady state response for Alzheimer's disease, and they have shown long-term memory effects because they're working on the issue of, okay, we have treatment, but this treatment does not improve the level of life experience, the life quality. They only prolong and slow down the process of the disease, right? And they have shown long-term effects due to the targeting of sharp wave ripples. And you have mentioned that you have found short-term memories, so you can keep more items, whatever they are, in your short-term memory at the moment. Would it be possible to combine these two methods? I mean, in the end, there could be the issue that you have a stimulus-driven oscillation, and then the oscillation that is evoked by the tACS, what might interfere with each other. But from my experience or my perception, it could be that the initial enhancing of short-term memory capacities could also contribute and thereby multiply the long-term effects. And this would be very beneficial to Alzheimer's disease patients, which are to this day still one of the most endangered patients in terms of life quality and also, of course, health.
Yes, again, a very good question, and I could give another talk on that. I'll try to keep it short. There's so many things that come to my mind. So you're completely right. Let me at first talk about what I think would be the most beneficial to improve long-term memory. There is the group of, I'm bad with names today, they have a sleeping lab in Tübingen, and they apply tACS during sleep, and they know which types of brain activity, like the delta activity during deep sleep, are beneficial for which type of memory. And they can selectively enhance different types of memory, declarative versus non-declarative, etc., etc. And that, you're right, could probably be combined with some of the things that have been used to increase short-term memory. And what we did is not the only one. There were also nice studies by Walter Paulus group from Göttingen, where they had nested gamma oscillations in certain phases of the theta cycle in parietal and frontal regions to mimic what they find there in the EEG while people do a short-term memory task. And as you said, if you combine that, the short-term memory tACS during the experiment and then the nighttime tACS to enhance long-term memory, that would probably double the effect or potentiate it, I don't know. But regarding Alzheimer, because you say that, there is a lot of literature on using tACS, on using 40 Hertz stimulation that started out in a mouse model of Alzheimer, where they made the mice ill. I don't know how you can induce that, but they had the amyloid beta plaques and the tau fibrils and they actually showed two things: that the 40 Hertz stimulation reduces the number of these Stoffwechselendprodukte, of the bad stuff, the waste in the brain, and they improved in performing in a maze, which is some kind of memory task. And this is being applied now by many groups. If you look this up on clinicaltrials.gov, you can see that everybody's experimenting with 40 Hertz oscillation, and they flicker at people, and they do 40 Hertz auditory stimulation, and they do 40 Hertz tACS and 40 Hertz TIS in Alzheimer patients. And I expect that in the next 1 or 2 years, we'll see the results and see what works best.
Thank you very much. Okay. Yeah, I think at the moment there are no more questions, but if so, please feel free to just interrupt me. Yeah, but I do think that we can come to an end. And so thank you, Professor Herrmann, for this really, really nice talk. I personally really enjoyed it, and also you also got feedback from the audience already. I do think we all enjoyed it. And yeah, thank you all of you for your attention. Thank you for showing up. If you would like to have a more detailed look at the work of Professor Christoph Herrmann, we just linked a few of his online appearances, and also the most basic thing, his Google Scholar, so that you can look up his publications. And also, he offered it, if you're interested in modeling stuff, reach out to him, maybe a collaboration occurs. Yeah, and with that being said, I do think we're at the end of today's talk, and I would say thank you very much, enjoy the evening, and see you next time.
Bye.
Bye-bye.
Spot on 90 minutes.
Perfect.